through time in terms of their actual pedigree. You can specify who mated with whom and which offspring were produced from those matings. And this is one such computer-generated tree where I tried to draw males and females in each and every generation across 20 generations. And we had an ancestral population down here that was split by a vicariant event into two sister populations here. And this population was later split by another vicariant event into two populations. But what we're doing through here is just simulating an actual pedigree. And it's going to be through this pedigree that genes have been transmitted. So this next slide shows exactly the same pedigree, but now I've traced the matrilineal history by the dark arrows. So again, coalescent theory comes into play here. So starting from the current generation at time 0 up at the top, you can see from the heavy dark arrows how these different lineages coalesce. This is the matrilineal or the mitochondrial gene tree. So this is a gene tree, but it's within an organismal pedigree. It's a component of the organismal pedigree. It's not one and the same thing as the organismal pedigree. It is just a component of the species tree or the organismal tree. So that's a mitochondrial gene tree, the kind of thing we reconstruct for the matrilineal history. But one can also point out, point number 13, that this gene tree concept is very powerful and generalizable. So for example, let me now take that exact same computer-generated pedigree, and this time, instead of tracing the mitochondrial lineages through it, this time I'm going to trace the Y chromosome gene tree. We're thinking like mammals, for example, that have a Y chromosome. And if there are genes on the Y chromosome that have been transmitted through that same pedigree, they will have been transmitted from the male to male to male to male to male component of that pedigree, and that will define another gene tree within the same organismal tree. But you can see, if you remember, that this differs from the matrilineal tree that I just showed you. It has to, because it had a different transmission pathway. This is the patrilineal gene tree, the paternal analog of the matrilineal gene tree that was provided by mitochondrial DNA. So that's a Y chromosome gene tree. And one can imagine other gene trees as well. These would be for nuclear genes that are transmitted through both one sex or the other in each and every generation through the pedigree. So there's a very large number of different nuclear gene trees possible through that same organismal pedigree. And each one will show its own idiosyncratic tracing. They won't all be the same. They will be different from one another because they have different transmission histories in reality. This is not like just a conceptual theory or something. This is the truth. I mean, they will, from what we know about Mendelian heredity and the basics of genetics, there will be truly different gene trees, and there will be a multitude of different gene trees within any organismal phylogeny. There will be large numbers of different gene trees representing the different histories of transmission for various nuclear genes through that pedigree. So that leads me to point number 14, which is that in sexual species, but not in asexual ones, gene trees in principle are multitudinous and non-isomorphic. That is, they're not the same from one to the next. This would not, the only exception to that would be for asexual species. In any species that is truly asexual, all of the gene trees should be concordant because they will have the same history of transmission through the organismal pedigree. Which leads me then to point number 15, which is now taken for granted by everyone. But again, it was revelational when the field first began to think about this thing. Gene trees can also differ topologically from population trees and species trees. It is not enough that you have a gene tree that you can say you have a species tree, because different gene trees can differ and do differ topologically from one another. So there could be, here's a population tree with just one bifurcation in it, for an example. Some splits in the gene tree predate the population level split. Other splits in the gene tree might postdate the population split. So it's important to, again, recognize this distinction between gene trees and organismal histories. Here's a number 16 on my list. We're getting there. Remember, we're going to 22. So we're up to number 16 now, so you can keep track of where I am. And this is one that particularly intrigues me. And with respect to gene trees, the phylogenetic status of sister populations or sister species is itself an evolutionarily dynamic condition. It's not as if when a species tree splits that there's some fixed relationship of those species from that time forward. Instead, with respect to the gene trees that they contain, the phylogenetic status of those sister species itself evolves through time. So I tried to diagram that here on this slide, but the basic idea is that if you have an ancestral population back here, and if it gets split by a barrier to gene flow, if you were to look at that shortly after the vicariant event, shortly after the splitting event, it would be the truth in reality that some lineages within population A are actually genealogically closer to some lineages in population B than they are to other conspecifics. And the reverse is also true. Some lineages within B would be genuinely closer to some lineages within A than they are to other lineages within their own species. A shorthand way of saying that is that with respect to gene trees, two daughter species shortly after their formation are very likely to be polyphyletic with respect to their matrilineal or gene tree, the matrilineal histories or the gene tree histories. But that is not a fixed status. Through time, because of lineage sorting and coalescent processes and branching processes, those lineages will sort such that some go extinct where others spread within each of the daughter populations, so that after a certain amount of time, we might well expect to find a paraphyletic relationship where one of the populations, population A, is paraphyletic with respect to population B, or vice versa. But lineages continue to sort through time. Some lineages go extinct, others expand and become more abundant, so that branching process theory or coalescent theory shows that after enough time has passed, on the order of 4n generations, these daughter populations are likely to evolve to a situation where they are reciprocally monophyletic with respect to the gene trees that they contain. But this lineage sorting process is itself evolutionarily dynamic, so that a common time course of events following the speciation is that species may start out with respect to their gene trees being polyphyletic, but then evolving to a paraphyletic status, and then through time evolving to a status of reciprocal monophyly. Sometimes the polyphyletic stage can be bypassed if the speciation event took place on the periphery of a species range. Then you might go straight to a paraphyletic situation and then eventually sort to one of reciprocal monophyly. But these are time and demography dependent phenomena. So to me, it was mind-blowing to think that species could actually be evolutionarily dynamic in their phylogenetic relationship to one another with respect to the gene trees they contain. Point number 17 is also something that I'm sure all of you take for granted, but it was a revolutionary concept when it was first thought of. And this is the notion that intraspecific genealogy and historical demography are thoroughly intertwined. Indeed, you can't hardly talk about genealogy without talking about population demography. These are like two sides of the same coin. In order to understand genealogy, which is the history of how lineages evolve within and among species, it's inextricably related to the historical population demography of those same species. So there's lots of algorithms now for trying to reconstruct organismal history or demographic history from observed genealogies. Here's just one example from humans, where in the past there was a major population expansion. When populations expand very rapidly, lineage extinction is slowed for a while so that one can get a peak in the mismatched distribution of times to common ancestry. And that can be reflected in the gene genealogy with a clustering of nodes at the point of the population expansion. So there are ways to convert thinking about population demography to gene genealogies. But this whole concept that genealogy and demography are connected was revolutionary because traditionally phylogenetics, people that did phylogenetics were working above the species level, and they never took demography into account when making their species phylogenies. Perhaps they should. We now know for sure at the intraspecific level, you can't do phylogeny or genealogy without reference to extractive demography because the two are so tightly intertwined. Another point related to that is that in many species, key behavioral and demographic parameters can differ between the sexes. So, for example, if we plot female dispersal of gene flow on this axis and male dispersal of gene flow on this axis, we could see that there might be some species in which female dispersal is low and male dispersal, for example, is high, like that would be done in the lower left corner quadrant of this diagram. If such a species has low gene flow mediated by females, but is panmictic with respect to males, high gene flow by males, we would expect there to be tremendous population structure in mitochondrial DNA, but perhaps no structure at all in nuclear genes, which are transmitted through both sexes. But even the matrilineal component of the population history could be very important to know because female demography is especially important, the female component of a population's demography is especially important. If females fail to reproduce, the population would go extinct, so for conservation or other purposes, it would be especially important to know even the matrilineal history of that species, even though we appreciate that it's, of course, it's only a minuscule fraction of the total hereditary history of the species. It's an important component of that history because of its tight connection to female population demography, which is also often very important in conservation and other purposes. Which brings me to point number 19 then, that principles of what we call genealogical concordance can address the depth and strength of phylogeographic population structure. And I don't have time to go into any detail with this, but there's a whole field now that's addressing the concern with the concept of genealogical concordance. And I recognize 4 different components or aspects to this genealogical concordance. Aspect 1, 2, 3, and 4. Aspect 1 would be, for example, if you have studied just a single gene, such as mitochondrial DNA, within a single species, such as species 1. If you see a major break in that species, what that really means is that there would have been multiple nucleotide substitutions or changes that distinguish one phylogroup from another, and that would be recognized, for example, by high bootstrap support and a mini phylogeny for those populations. So you would see concordance across characters within the gene. That's aspect 1 of concordance. Aspect 2 of concordance would be that if you have that same species and now you look at a number of genes, it might be mitochondrial DNA, it might be one of those genes, but you might also look at a series of unlinked nuclear loci. If you see concordant patterns emerging across multiple genes, that would give you confidence that your gene trees are reflecting properly a split at the organismal and the population level. So that kind of concordance we call aspect 2. It's a multi-gene concept of concordance. Aspect 3 is a multi-taxon aspect or component of concordance. The basic idea is that you could be looking at one or more genes in multiple co-distributed species, like I was doing earlier with the Atlantic versus Gulf of Mexico populations of many species. If you find many species that are concordantly subdivided, that would be an example of what we would call aspect 3 of genealogical concordance. And again, that would tell you that there's something probably about those gene trees that's trying to tell you something about the historical demography of the populations in which they are embedded. And then finally, aspect 4 of genealogical concordance is to try to connect the data from molecular genealogies with organismal information. This might be, for example, information about the vicariant history or the geological history of an area. If you see concordance between what you expect might be happening and the structure of genealogies within a species, that would be what we call aspect 4 of genealogical concordance. And that helps to emphasize the point that whenever you're doing phylogeography, it's important not only to get your molecular genealogies, but also to interpret the data in the context of whatever you might know about the geography or the history of a region, because this will inform the overall biogeographic scenario that you come up with. Which brings me to point number 20. Hang in there, we're almost done with these points. So This is probably also obvious to you, but it wasn't necessarily obvious in the early days, but now it's taken for granted. And that is that phylogeographic findings can have key importance for taxonomy and conservation. I'm sure we will hear much more about this in our talks at these meetings, but it should be obvious really in retrospect that phylogeographic patterns have huge relevance for taxonomy and for systematics. And I'll just mention one example. In my lab, we spent about 10 years working on all the species of marine turtles. And we worked out the phylogenetic relationships of these turtles and the phylogeographic patterns within each of these species. And it had tremendous relevance for the taxonomy of these creatures, as well as for conservation purposes, because these are all endangered species or threatened with extinction. And so the phylogeographic findings had tremendous relevance to taxonomy and conservation. So that brings me to my final 2 points, numbers 21 and 22. And these are the most important of all. If you forget all the other ones, don't forget them all. These final 2 are what I would say are the most general, the most lasting contributions that the phylogeographic revolution has made to evolutionary biology. And the first of those 2 points is that phylogenetic reasoning is relevant at the intraspecific level, as well as at the interspecific level. When I was in graduate school, I was taught that phylogeny had no meaning at the intraspecific level, because that was within the realm of lineage, anastomosis, and particulate evolution, because of sexual reproduction, connecting all the pathways within a species. So I was taught that phylogeny is a supraspecific concept, something that phylogeneticists did above the species level, but not within species. But the phylogeographic revolution has made it abundantly clear that we can think phylogenetically or genealogically, even within species. It's relevant to do this. It's not only relevant, it's very important. And it connects with phylogenetic reasoning above the species level as well. So that was one of the really major contributions, I think, of phylogeography, which then leads, so we can think then of phylogeny on a macro scale as being a branching tree, such as like that. But if you were to look with a closer magnification at any of the branches in those trees, ultimately, and look closer and closer and keep looking closer and closer, ultimately, those would be reflected or resolved as population pedigrees. And it's through those pedigrees that genes have been transmitted at the microevolutionary level. So phylogeny at the micro and the macroevolutionary level are ideas on a continuum. There is no clean demarcation between these two. These are ideas on a continuum. Which leads me then to my final and perhaps most important point, and that is that microevolution, just like macroevolution, is inherently historical, and it can now be studied in explicit genealogical, that is to say, phylogenetic terms. So if we're to divide, we sort of do a summary slide here. We've got the microevolutionary disciplines within species. We have behavior, population demography. We have traditional population genetics. And then we also have the macroevolutionary disciplines of historical geography, paleontology, phylogenetic biology. When I was in graduate school, these two arenas, microevolution and macroevolution, were largely distinct. There were very few connections between them. You could be trained as a phylogeneticist, which meant that you worked above the species level. You would probably be trained in a museum somewhere. You would learn the language of phylogenetics. You would know what a synapomorphy is and distinguish it from a symphysiomorph. And you would talk about clades, and you would talk about phylogenetic units, monophyly, paraphyly, polyphyly. All of these terms would be familiar to you if you were trained as a macroevolutionary biologist or a phylogenetic biologist. Conversely, you could be trained at the within species level, in which case, like me, you would have been a population geneticist. There you would have learned the language of population genetics, which is that you would be dealing with allele frequencies, and you would be studying how mutation, migration, selection, drift, and mating system can influence the allele frequencies and genotype frequencies within populations. But you wouldn't necessarily have ever heard anything about phylogeny because it was not a concept that was applied at the intraspecific level. So there was this gulf, this major distinction between the macroevolutionary disciplines on the one hand and the microevolutionary disciplines on the other. I think the most lasting contribution of phylogeography will have been to form a bridge between these two disciplines. Phylogeography is right here in the middle. It connects the macroevolutionary disciplines to the microevolutionary disciplines. These are all now can be thought of as ideas along a phylogenetic continuum ranging from the micro to the macroevolutionary. And of course, it also brought all the molecular field of molecular genetics into the picture as well. So it connects with phylogeography. So phylogeography, in my estimation, will have its most lasting impact in trying to unite the two major branches of evolutionary biology into one more coherent whole. And I hope that all of you students that invited me to these meetings will carry on and with this tradition, and will go on in your careers, and it'll be very interesting in 20, 30 years from now to see how fully integrated the fields of micro and macroevolutionary biology will become. I want to thank you very much for your attention.
Muchas gracias al doctor Eli por acompañarnos por su plática. Si gusta, bueno, ahora tenemos tiempo para un par de preguntas. Si alguien tiene alguna, puede levantar la mano y nuestros compañeros les pasarán los micrófonos. Questions? Any questions?
Hey, John. So when you I'm sorry. John, when you published your major paper in 78 or 79, why did you pick the Florida Naturalist of all journals?
That wasn't the Florida Naturalist. The paper appeared at PNAS.
So
That was just a popular article we wrote about bookers.
Oh, okay.
That was where the picture came from.
Okay.
The paper, the science paper was.
Right. Okay. I thought maybe you did another paper in.
No, sorry. The original papers were in genetics and PNAS.
Hi, John. Considering all the experience you have and all the tasks that you have seen, and mentioning the fact that phylogeography also helps in taxonomy and all these classical disciplines, what would be your take in this issue regarding phylogroups as a species or subspecies? And just because that's still alive in literature, what patterns have you seen regarding, the phylogenies, genetic distance between the two phylogroups and all the sort?
Of course, this is a big question. The question was, what is the relationship of phylogroup to what might be called subspecies? Actually, I won't say to say anything about this because I realize it could be a very contentious area. But personally, I feel like the term subspecies it's existed for a long time, and it could just as well be used in the context of phylogroups. In other words, phylogroups could be more or less equated with subspecies in an ideal world. But the major point is not what we call things, but to just appreciate what the whole phylogeny or structure is, and to appreciate that there can be dramatic structure within what are considered taxonomic species. Whether we call these subspecies or phylogroups is a matter of taste. But the important point is to appreciate that these entities do exist. There's a history there that should be tackled.
And in that question, going back, and what about genetic distance in separating taxonomic entities?
Again, these are ideas on a continuum, but I tend to think of if things differ, for example, in mitochondrial DNA, I tend to think of things being in different polygroups, probably if they're on the order of, say, 1% sequence divergence or more, something like that, as being an arbitrary cutoff point. But these are ideas on continuums. So, of course, when we talk about polyreview, if you talk about mitochondrial DNA, you always have to be aware that you're just talking about the gene tree. So before you want to recognize subspecies or something in a more formal taxonomic sense, you would presumably want to have concordant support for that from the nuclear genome as well.
Thank you.
¿Alguien de las personas que nos escuchan por videoconferencia tiene alguna pregunta o por el canal de YouTube? Bueno, pues entonces ahora sí, agradecemos a los tres. En este momento tenemos tiempo para un descanso. Esperemos regresar puntualmente y ahora afuera se tomará la foto del grupo, por lo que les pedimos se coloquen frente a la entrada donde estarán los fotógrafos para tomarlo. Gracias.
El receso tendrá una duración de 20 minutos. Regresaremos aquí a las 11:20. Aquí los esperamos.